This material is made available to you by or on behalf of the University of Melbourne under Section 113P of the Copyright Act 1968. It may be subject to copyright. For more information, visit the University Copyright website.
Anyone has any question before I start from last week, from the prac? Any general question? If not, we'll continue to talk about microbial hazard. I believe we stopped here last week when we start talking about different type of microorganism and what type of hazard associated with each group of microorganisms. And we touched briefly on the difference between accidental versus obligate and opportunistic. And in fact, all of these are very risky, very dangerous, no matter if it is accidental, opportunistic, or obligate. But in terms of how does it happen, we said, for example, the accidental is the best example is Clostridium tetani, which very widely present in soil, and usually when farmer or any person walk barefooted in the farm, and if they have wound or cut on the foot, this microorganism can penetrate through the cut or puncture on the foot, and consequently cause a problem. The obligate from the title, meaning it is present under a special environment, and I mentioned Neisseria as the best example, which usually present in those people infected with Neisseria gonorrhea in the urinary tract. And the third one, the opportunistic, we said, again, based on the title, opportunistic, it means it's not obligate. And usually it is associated with those people who have depressed immune system. So they are more sensitive than regular person because the immunity system in those people is weak, and infection can be done through skin infection or through respiratory system or even through direct contact with other people who carry the bacteria or the virus. And the best example is Pseudomonas aeruginosa. So that will lead us to talk about virulent microorganism and toxin. I did mention also before that when we talk about toxin, the problem with toxin, they are colorless, they are smellless. You cannot smell it, you cannot see it, you cannot detect it. But if it is present, it can cause what we call it acute toxicity, meaning it can cause sudden illnesses, which can be very serious depending where that toxin comes from. Is it from Clostridium, Staph aureus, or from mold like Aspergillus, as we talk and discuss later on. So when we talk about virulence, we have to distinguish between different terms. Infectivity, which means the ability to establish contact point or focal point for infection. Invasiveness, meaning the ability of the pathogen to invade the targeted objective and spread. And in a minute, we'll talk about the method that pathogen or toxin is transferred to human and how it invade and spread. And after invasion, then it will start to show the toxogenicity or the outcome of that infection, meaning the symptoms associated with infection. Generally speaking, the virulence and the effect of toxin and pathogenic microorganism can be measured by what we call it LD50. And by definition, LD50 is the dose of any toxin or any chemical that's responsible for virulent effect that can kill 50% of the targeted population. What do we mean by that? Suppose you have 100 rats and you are testing a special toxicity of a given chemical in 100 rats and you use different doses. So the dose which kill 50% of them, meaning 50 will be killed, 50 remain alive, that's what we define it as LD50. So please do not confuse LD50 with MIC, which we discussed before, or in other words, what we call it the minimal inhibitory concentration. Remember we mentioned MIC, or the minimum inhibitory concentration. Minimum inhibitory concentration is the minimum concentration that can inhibit some microorganism from growth, and usually use it to test antibacterial agent. So LD50 is the dose of the toxin that can kill 50% of the targeted population. Now, how do pathogen transferred into the targeted host, no matter if it is a human or animal? So there are 4 stages: transmission, attachment and colonization, invasion and growth, and evasion and damage. So what do you mean by transformation and how those pathogens are transformed? Remember we said before, microorganisms are everywhere, in air, in water, in food, and because of that they can reach the targeted objective in different way. Like viruses, for example, can be transferred when someone is already infected or suffering from cold. You are on a bus or on a train. If he or she cough, the virus can be transmitted very easily to people surrounding that infected person. And the best example about this airborne virus is Mycobacterium tuberculosis, which can cause high fever, especially in kids when they are infected. Direct contact from person to person, and usually this is again more linked to those people who are suffering from depressed immune system, or we call them people with AIDS. Using a vehicle like vomiting, mucus, using a carrier or vector-borne, example, by fly, by ticks. So all of this can be used as a tool. Water could be another vehicle for transformation. Sometimes we may classify food as a vehicle. Anyone remember the best example about food being a vehicle for transmission of the pathogen or the virus? I did mention before the example about strawberry. We said the strawberry was infected with virus, and in that case, strawberry becomes the vehicle for this transmission or transformation. So after the pathogen is transferred and reach the target, they want to establish themselves. So they usually attached to the mucosal surfaces in the nose, in the respiratory tract, and quite often they try to build biofilm and try to spread very rapidly, as we'll talk about the third option. So when we talk, for example, about, I mentioned here, Vibrio, cholera, Cronibacterium, diphtheria, these are examples of very nasty pathogen which can infect the respiratory tract in a human and animal and cause severe problem. So after they have established the contact, they build the biofilm, we have to mention a little bit about evasion and growth. And please here, make sure you distinguish between invasion and evasion. Invasion meaning they try to grow and spread in large number, so they don't stay with their number. Like if even a few cells reach a contact point, nose or mouth or any part of the body, skin could be sometime in people with depressed immune system, they try to invade and increase in number. However, those people who have a strong immunity system, they can develop barrier and prevent those microorganisms from spreading. So that will lead us to evasion of host defense. So if the microorganism can avoid this barrier, they again can spread and accumulate. But if a person has strong immunity system, that strong immunity system will prevent the microorganism from passing that barrier. Many bacteria exhibit phase variation once they are in the body. What do you mean by that? Sometimes microorganisms may change the shape, the resistance for the host or the surrounding environment as far as they established themselves inside the host. They could, for example, produce a spore if they have the ability to sporulate, so they can stay for longer time. And as a result of all these steps, transformation, attachment, invasion, they then either produce toxin and show the system in terms of intoxication, or if it is a gram-negative bacteria, they cause problems through what we call it infection. And we'll distinguish between infection and intoxication in a minute. But no matter if it is infection or intoxication, the final result will be toxogenicity, meaning causing problem related to the toxin produced by that pathogenic microorganism, no matter if it is gram-positive or gram-negative bacteria. So that leads us to what are toxins, what is the different type of toxin, where do toxins come from, are they limited only to bacteria? And definitely the answer is not, as we'll see in a few minutes. But the common toxin that we usually discuss when we talk about food safety is usually bacteria related, because bacteria, especially spore-forming bacteria, are the most dangerous microorganism when it comes to food safety. Saying that, I don't mean to pass the message that other toxins from mold or from algae, for example, are not important. They are important and dangerous, but they are less frequent because of the nature that the food comes from. So when we talk about algae, for example, later on, or mycotoxin, you will understand the difference between bacterial, myco, or algae toxin. So as a start, we have to distinguish between two types of toxin, endo and exotoxin. So exotoxin, as you can sense from the meaning, exo meaning outside, meaning the toxin will be produced and released outside the cell. Endotoxin remain inside the cell. And usually endotoxin are associated with gram-negative bacteria, meaning they will be released when the bacterial cell lies, die, and release the toxin because it is inside the cell membrane. While exotoxin, the microorganism can grow and release the toxin outside. And the best example will be Staphylococcus aureus, Bacillus, or Clostridium botulinum. They all classified as exotoxin. Again, endotoxin, as I mentioned last week, the chemical nature is mainly lipopolysaccharide with some protein molecule. Usually it has very complex chemical structure. So now we group toxin into endo and exo. Now, where does the toxin come from? So aside from where does it come from, we say the major two types of toxin are endotoxin and exotoxin. But where do they come from? They can come from bacterial sources. We call it bacterial toxin. They can come from algae, algal toxin, or they can come from fungi, and we call them mycotoxin. And we'll talk about each of these groups in more detail. To start with the bacterial toxin, again, when we talk about bacterial toxin, we face with the major two types, exo and endo, because under bacteria, we have gram-positive and gram-negative. So gram-negative usually associated with endotoxin, gram-positive exotoxin. However, when it comes to the exotoxin, again, we can subgroup the exotoxin to three major subgroups, I call it, and that depends on what type of effect they cause. Namely, enterotoxin, and enterotoxin usually infect our digestion system, so it can cause dysentery, diarrhea. And please don't mix between entero and endo. So endo meaning remain inside the cell, entero refer to the location where the toxin is released. It is in the gut, so we call it enterotoxin. If it is in the nerve system, we call it neurotoxin. It can infect the brain of animal and the human. And the best example about neurotoxin is Clostridium, especially the one we mentioned before, which cause tetanus, Clostridium tetani. Remember, we said it can get into the human body through a cut or puncture on the foot or cut skin, because it's present very widely in the soil. So Clostridium tetany can be transferred very easily from the soil. The third group of exotoxin is what we call it cytotoxin. And cytotoxin interfere with the RNA or the ribosomal function, which is responsible for protein manufacturing, and it affects the protein synthesis in the targeted individual, no matter if it is animal or human. Any question about the difference between entero, cyto, or neurotoxin? So again, I emphasize they all different type of exotoxin, and exo is different from endo because it is produced and released outside the pathogenic microorganism. Endotoxin, as we said before, it is associated with gram-negative bacteria. In nature or chemically, it is made from lipopolysaccharide and protein. And I listed here example of gram-negative bacteria. So whenever we say gram-negative, you should come to mind Salmonella, E. coli, Klebsiella, all of these are gram-negative bacteria. And the most dangerous one, in fact, they all bad, but Salmonella and E. coli 0157 is very toxic, very dangerous gram-negative bacteria. The endotoxin can cause fever, and usually the fever is a symptoms of this intoxication, because in our body, what we call it the white blood cell, or the polymorpha nuclear leukocyte cell, build a defense mechanism against any foreign matter. So when the toxin reaches the body, enter the blood cycle, a lot of these polymorphonuclear leukocyte cell, or what we call it the white blood cell, try to attack and stop that toxin from spreading. Consequently, special messages will be sent to the brain, and as a consequence, the temperature of the body will increase. So the increase in temperature is an indirect indication about the interaction between the defense mechanism by the polymorphonuclear, or the white blood cell, and the invade toxin. So after this brief introduction about the different type of toxin, endo, exo, and the different group of exotoxin, depending on the symptoms it causes, being entero, cyto, or neurotoxin. Now, there is another classification about food poisoning, depending on how does it happen and what causes that food poisoning. So that leads us to another term which we call it infection and intoxication. So infection is usually associated with the gram-negative bacteria, and intoxication is associated with the gram-positive bacteria. Anyone has any guess why infection will be associated with the gram-negative bacteria, while intoxication will be associated with the gram-positive bacteria? Eva? Sorry? No. Toxin? Yeah, because exotoxin is formed by gram-positive bacteria, not because of the spores. Because here we are talking about disease-causing agents, so we are talking about the toxin, not about the spore. Remember, we said the spore, if it is dormant spore, it could cause no danger at all. But the danger will appear when the spore sporulate and goes back to vegetative state. But the spore as is not dangerous. So infection is associated with gram-negative bacteria, intoxication associated with gram-positive bacteria, because gram-positive bacteria are usually classified as exotoxin or associated with exotoxin because they produce the toxin and release it outside the cell. And when we discuss the example about infection and intoxication, we'll notice that infection is linked to gram-negative bacteria, intoxication related to the toxin itself. And because the toxin is coming from gram-positive bacteria, so intoxication is linked to gram-positive bacteria. So as summarized again in this slide, infection caused through the consumption of food containing gram-negative bacteria. Because as we said, it is not free toxin, it is the whole cell of microorganism. So as you digest food contaminated with E. coli 0157, when the E. coli reach the gut, it will be hydrolyzed and as the cell die, it will release the toxin from the cell membrane and start to cause the symptoms. While intoxication, we are talking about the toxin or consuming food already contain the toxin. So if you have, for example, canned meat sample, you open the can, you didn't smell any bad smell, you ate the meat, and if there is any Clostridium which has spore, the spore germinated, produced toxin inside the canned meat, that toxin can cause intoxication because of the toxin already released into the meat sample. I emphasize again, as we mentioned last week and at the start of this lecture, toxins are odorless, tasteless, colorless, and that's why it is hard to detect. So that reminds me with the statement we said last week also, not every spoiled food is dangerous, but again, not every normal food is safe. Remember this statement we said last week? We said not every spoiled food is dangerous, and not every normal food is safe. What do you mean by the first part of the sentence? Not every spoiled food is dangerous. We said if it is spoilage only, spoilage microorganisms are non-pathogenic, so it doesn't cause disease. So if you eat, for example, a cheese which has high acidity due to the growth of lactic acid bacteria, it will be very sour, but not necessarily causing problem. But if we talk about the second part of the sentence, not every normal food is safe, that's related to the toxin. When we say not every normal food is safe, meaning if the normal food contains toxin, it won't be safe to eat because toxin is odorless, colorless, tasteless. So we have to be vigilant about the type of food we eat, no matter if it is normal or if it is a spoiled food. So as I said before, we'll cover a few examples about infection and a few examples about intoxication. So we'll start with infection or foodborne disease caused via infection. And again, keep in mind when we talk about infection, we are talking about food poisoning related to gram-negative bacteria. So the first example is salmonellosis, and from the title you can guess it is caused by salmonella. And because salmonella is a gram-negative bacteria, the disease associated with salmonella is named salmonellosis. Usually salmonellosis is associated with poultry product, egg, chicken meat, because salmonella can be very widely present in poultry product. I'll pause here for a few minutes. We have William, a master student. He is in his second year preparing a special project, special food sample for his project. He would like to make a short announcement. William?
Hi everyone, I'm William, and as you probably, some of you have probably heard, I'm going to have like a sensory analysis today, and on my lupin sausage, this lupin sausage. And in case you don't know what lupin is actually these flowers right here that you see plenty of them in New Zealand and Western Australia. And what we're actually trying to do is like to incorporate this lupin flower into the emulsion system to the sausage and see how it affects the texture, the flavor. So the sensory analysis will take place at a food sensory lab, building 184 from 2 to 5 p.m. No bookings required. You can just come in and try it, and probably only take 10 to 15 minutes of your time. You just need to try out 6 different formulations. And as long as you eat meat and you have no allergies for gluten, wheat, soybean, and nuts, you can just come in and try it. And I guess that's it. And I hope I can see you guys later.
Okay, William. So anyone would like to help and participate between 2 and 5? Sensory test. So you test different type of sausages fortified with lupin. Again, when you run sensory test, you don't have to swallow the sample. You can test and spit it out. It is done on the ground floor in building 184, between 2 and 5, correct, William?
Just come in, no bookings required.
Okay. Is there any incentive if they participate?
There's some chocolates.
So he will give you some chocolate if you participate. Okay, good luck. So coming back to salmonellosis, again, with all the example I mentioned about infection and intoxication, I don't expect you to memorize all the detail, because when it comes to the symptoms, for example, like increase in temperature, vomiting, diarrhea, headache, they are similar in all food samples, but usually the incubation time vary from microorganism to microorganism. But you have to keep in mind that when we talk about infection, we are talking about gram-negative associated diseases, intoxication gram-positive. So with salmonellosis, for example, the incubation time 6 to 48 hours. Why it is so wide range in terms of incubation period? 6 to 48 hours. Why don't we say 6 to 8 hours? What cause or the reason behind such wide range from 6 hours to 2 days? Anyone? Sorry? Incubation? The immune system? What do you mean? Suppose all people are adult with the same immune system. Again, the period is still from 6 to 48 hours. I don't want you to memorize statement as is. You have to think the logic behind it. What other possibility? The? No. Sorry? Related to the toxin? We are talking about the gram-negative bacteria, so we are talking about the whole cell. Remember, we said with the gram-negative, the toxin inside the cell wall. So we are not talking about the toxin. Exactly right. The initial inoculum of the bacteria, how badly the food sample is contaminated with the salmonella. Was it 25 colony? Was it 100? Was it 2,000? So badly contaminated sample will have short incubation period. Less contaminated sample will have longer period. And that, in fact, again, represent some of your results. You noticed yesterday, some of you, when you checked changes in the color, when you check the indole test, you notice different intensity in the pinkish color. And I mentioned to some of you that the variation in the intensity depends on the load of the inoculum you took when you transfer the culture from the Petri dish to the slant using the loop. So these variations are related to the size of the infection negative bacteria, or in your case in the lab, the size of the inoculum when you streak the biochemical reagent. How do we prevent salmonellosis? Again, if you are asked about prevention of specific, against specific microorganism, first of all, you have to think, is it gram-positive or gram-negative? Generally speaking, gram-negative are easier to kill because they are non-spore former. Gram-positive are more difficult to kill because most of them are spore former. So you can see here, normal cooking can destroy and eliminate Salmonella because it is a gram-negative bacteria. It can be killed by normal cooking. Can we eliminate Salmonella by freezing or refrigeration? All microorganisms cannot be eliminated or killed by refrigeration or freezing. They can be suppressed, so they stay dormant, but you cannot kill them. You can kill worm, protozoa, as we mentioned before, because they are larger in size, and due to the formation of ice crystal, they can be killed by freezing. But bacteria cannot be killed by freezing or refrigeration. The second example of infection, again, another gram-negative bacteria, Shigella sonnei. You can see the characteristics are similar to Salmonella. It is facultative aerobic. It is a gram-negative rod. Symptoms are similar. You can see here the incubation period is 1 to 7 days. In Salmonella, we said the incubation is between 6 to 48 hours, 6 hours to 48 hours. Here it can go up to 7 days. You can use the same analogy and say that this can be related to the strength in the immunity system of the targeted person, as well as the infection dose when it comes to the food that has been consumed, how much Shigella present in that food sample. Another remark about infection in general, they are mostly associated with fecal contamination. Most of the Shigella, Salmonella are associated with fecal contamination. So it could be, for example, through using badly contaminated irrigation water, or fecal sewage reach the sample in one way or another. So as mentioned here, people excreting Shigella through fecal sample could be the main source of this type of intoxication. The food involved, similar to the Salmonella, it involved also poultry in general, but in this case we added macaroni, salad, lettuce, moist and mixed food. Again, you don't have to worry about these details. The third example is listeriosis. Again, the main difference here, listeriosis from the title, similar to salmonellosis. Listeriosis is related to Salmonella. But in this case, and similar to Salmonella and Shigella, keep in mind that Listeria is gram-positive. It is not gram-negative. But again, because the toxin is not released outside, remember we said before, gram-positive usually excrete the enzyme outside. We call it exoenzyme. But some of them, like Listeria, still classified as infection because the enzyme is not released outside. Listeria in general, whenever you talk about listeriosis, because it is psychrophilic microorganism, it is usually associated with food that are kept refrigerated. So probably the second slide mentioned the food. So as you can see, mostly raw vegetable, dairy product, milk, and all these products usually stored refrigerated. Because they are stored refrigerated, if there is any contamination with the Listeria, especially Listeria monocytogenes. It could cause severe problem, like it can cause miscarriage in a pregnant woman. Infant can be killed. Again, that's related to the immune system in these targeted people. So 3 examples of infection now. Another 3 examples of intoxication. Remember, we said the main difference between infection and intoxication. In intoxication, we are talking about toxin present inside the food, which means it is definitely associated with the gram-positive bacteria, which has already grown in the food sample and released the toxin. So in infection, we were talking about food containing the microorganism itself. And as you digest the food, the microorganism cell wall will be lysed, and the toxin will be released inside the gut. Here, the toxin already present inside the food. And you have to keep in mind, toxin in general are very heat resistant. Similar to the spores, regular heating cannot kill spores and cannot kill toxin. So I repeat again, regular cooking cannot kill spores or destroy toxin. The first example is Staphylococcus aureus. Some of you showed me microscopic slide yesterday about a gram-positive cocci or short rod, which could be related to Staph aureus, especially if it is coming from Baird Barker agar or mannitol agar, because these media are selective for Staph aureus. General feature about Staph aureus, as I mentioned before, they are very osmotic tolerance, meaning they can tolerate high salt content. And that's why, as I mentioned previously, they can cause a problem in feta cheese, because feta cheese is high salt cheese. Again, you can see the symptoms are similar: nausea, vomiting, diarrhea, dehydration, stomachache. Incubation period, 1 to 6 hours. Generally speaking, when you run comparison, you will see that the incubation period for intoxication is shorter than infection. And this could be a good question for the final exam. So I may ask you why the incubation period for infection is longer than the incubation period for intoxication. Give some example. So if we go back to the incubation period for Shigella, we said 1 to 7 days. We mentioned Salmonella 1 to 2 days. While here in intoxication, we are talking about 1 to 6 hours. If I have another one, botulism, 20 to 36 hours. Generally speaking, if you are asked such a question, you have to say that because the toxin is already present inside the food sample, meaning whenever the food sample reach your gut, the symptom will start to appear. While in terms of infection, it may take some time for the microbial cell or the gram-negative cell to be digested and lysed inside the gut, then the endotoxin is released. So in another word, the release of the endotoxin will take longer time, and that's why the incubation period usually is longer than intoxication. But both cases, again, you have to say, depend on the infection dose, being number of cell or amount of toxin. So we carry Staph aureus, but we are not affected with Staph aureus if it is on the skin or in our nose. But as far as it reaches the gut through the digestion system, it can start to cause problem. The second example of intoxication is caused by spore former bacteria. And when we talk about spore former, we have to mention either Clostridium or Bacillus. So under Clostridium, again, we distinguish between proteolytic and non-proteolytic Clostridium. And from the name, proteolytic refer to the digestion and hydrolysis of a protein. And usually proteolytic Clostridium lead to the release of bad smell, usually in the form of hydrogen sulfide, which has really bad smell. And the best example, if you ever recall, a smell of rotten egg, that smell is coming from hydrogen sulfide. Usually Clostridium, as I mentioned before, associated with meat product. Meat and even egg, because it's very rich in protein. So proteolytic, meaning it can hydrolyze the protein. Non-proteolytic, it does not hydrolyze protein. This is similar to what we mentioned before about fermentative versus non-fermentative. But usually when we talk about fermentative, we are talking about sugar hydrolysis, not protein hydrolysis. So please do not confuse yourself between the two. Fermentative, talking about sugar hydrolysis. Proteolytic, talking about protein hydrolysis. So I listed here Clostridium perfringens. In the past, we listed Clostridium botulinum, and we have Bacillus cereus. So all of these are gram-positive bacteria and spore former bacteria. The difference between the two groups, Clostridium and Bacillus, anyone remember the difference between the two groups?
One's anaerobic and the other's anaerobic.
Correct. Which one is anaerobic? Yep. Yeah, Clostridium is anaerobic, as Eva said, and Bacillus is aerobic. So when it comes to your report, when you have to break it down, you have to go through these criteria: aerobic, anaerobic, rod shape, Kokai shape, catalase, oxidase, and so on. So Clostridium botulinum, as we mentioned with the Clostridium tetani, similar to this, it affects the nerve system and usually associated with headache, vertigo, double vision, and so on. Incubation period up to 36 hours. Botulinum come mainly from soil, water, and it's present in the intestinal tract of animals. It is mentioned here that it involves low acid food. Anyone remember the difference between low acid food and high acid food? What is the breaking pH? 4.6. Or to be more conservative, you can say 4.5. So any food sample with higher pH than 4.6, we call it low acid. Lower than 4.5, we call it high acid food. And we can use this as a barrier because Clostridium cannot grow in high acid food. Like tomato juice, for example. Tomato could have a pH close to 4.2. So we don't face a problem in tomato juice related to Clostridium because it is acidic. Similarly, when we talk about fruit juices, if it is a fruit juice with pH less than 4.5, we don't worry about Clostridium. And that's why sometimes, in fact, all the time with high acid food, we use pasteurization. There is no need to use commercial sterilization. So again, you can read through the feature of Clostridium perfringens. It is anaerobic, spore former, non-motile. It comes from soil. It causes infection through toxin, and so on. Bacillus, again, it is similar to Clostridium. Again, as we said, it is classified as aerobic or facultative anaerobic, while Clostridium, it is strictly anaerobic microorganism. But both of them are spore former. Again, Bacillus could be a major problem when we talk about the milk powder industry because it is a spore former. And if the cell start to form spore in the milk powder, it can cause problem. Again, as you notice here, generally speaking, it has very short incubation period, 30 minutes to 5 hours, depending on the dose, but definitely it is shorter than the incubation period of infection microorganism. Any question about the first group, which is bacterial toxin? So to recap, under bacterial toxin, we distinguish between endo, exo. We said exotoxin could be entero, could be cyto, could be neurotoxin, depending on their targeted damaged organ. We said example about bacterial toxin, we distinguish between infection and intoxication. Infection is linked to gram-negative bacteria in general. Or to be more specific, it is related to food which carry the bacteria inside. Because remember, we said Listeria monocytogen is classified as infection, even though it's gram-positive bacteria. So you have to be careful about this statement when we say, in general, it is associated with the gram-negative bacteria, meaning some gram-positive may go under that umbrella. And the best example is infection caused by Listeria. While intoxication, generally speaking, caused by gram-positive bacteria, because most, again, not all gram-positive, most gram-positive bacteria can produce toxin. And we listed Staph aureus, we listed Clostridium, we listed Bacillus as examples. We move to the second group under toxin. Remember, we said we'll cover bacterial, algae, and finally mycotoxin. So for algal toxin, we listed as number 2 because it is dangerous, but it is not as dangerous as bacteria. We don't hear too often about food poisoning related to algal toxin. But in fact, there have been some reports that about 10% of the foodborne illnesses in the USA is related to algal toxin. Because algae can be present in water, and usually fish, shellfish, can feed on algae, and if that algae is toxigenic algae, when a human consume or eat that fish, which already carrying the algal toxin, the toxin can be transferred to the human being. And it has been reported in the literature, in fact, the increase in the algal toxin in recent years is linked to the climate changes, because the temperature continue to increase, and that will again encourage the algal or the toxic algal to proliferate and increase in number. So as I said before, it has been reported about 10% of the foodborne illnesses in USA are linked to algal toxin. Luckily, here in Australia, we don't have incident related to algal toxin. Most of the foodborne illnesses are bacterial related. So the origin of algal toxin is usually marine algal toxin, meaning coming from fish, shellfish. The toxic species are normally present in low concentration with no environmental or human health impact. So generally speaking, they present in low concentration, and that's why we don't hear or face too many problems related to algal toxin. So consequently, the toxicity will be related to the concentration. So if it is low concentration, more likely it doesn't show the symptoms. So it is, in another word, less toxogenic as compared to bacterial toxin, especially Clostridium, which is more toxic even in minor quantity. For algal, there should be, or in another word, the LD50 will be much higher. A photo which shows the algae, so you can see that it is unicellular. It appears as a single cell, and it appears in different color. In fact, it has been reported in the Bible that the phytoplankton, or what they reported, the blood red plague, referring to the Red Sea. So the Red Sea is related to this phenomena. They call it blood red, which is related to the red color coming from special type of algae, because algae can come in different color, green, yellow, blue, and red. And in fact, it is well known that the Red Sea does not support the growth of any type of fish because it is very toxic. And sometimes it referred to as harmful algal bloom, or abbreviated HAB. So HAB, or harmful algal bloom, refer to the red algae in the Red Sea. And similar to bacterial toxin, again, out of about 4,000 known phytoplankton related to algae, only 2% are known to be toxic. But even it is 2%, we should not ignore it, and we should give it enough attention as a possible poison or intoxication for a human. Similar to the classification of bacterial toxin, again, microbiologists have grouped algal toxin into two major categories: dianoflagellate and diatom algal. So all the toxin-producing algae belong to either one of these two groups, dianoflagellate or diatom. Different type of algal toxin, again, don't worry about the name. Saxotoxin, brevotoxin, Siago, Ocadia, domic acid, all of these are algal toxin. Just remember the previous stain, the diato and the second group of the toxin, the dianoflagellate and the diatom. So again, under taxonomy, they mentioned saxotoxin, which is one of the 5 toxins present in the previous slide. It can come from 3 different genera, which belong to the group dianoflagellate, and these genera are Alexandrium, Geomondium, and Bromondium. Again, I'm not going to question you about these names. You don't have to worry about them. Just remember that the 2 major groups of algae that are toxin-producing are dianoflagellate and diatom. But don't worry about the different name and the different genera. Any question about algal toxin? So I repeat again, it is dangerous, but it is not as dangerous as bacterial toxin. because we are talking about 2% out of total number of 4,000 different type of algae. And usually it is associated with seafood. So the fish will eat the algae. When we consume the fish, we face the algal intoxication. Any question? We'll have 5 minutes break because before we talk about mycotoxin. Usually mycotoxins are not as dangerous as bacterial toxins. The same reason I mentioned for algal toxin. They are there, but as we mentioned before, because mold can be killed very easily as compared to bacteria, as we kill the mold, we eliminate the toxin. But for some reason, if the mold produce and spread in large quantity, it can cause serious problem. And in fact, if you read in the literature, it has been reported like in the 1970, 1974, there were major food outbreak in India. And it was related to the consumption of corn contaminated with mold. And the report indicated that about 400 people were infected and 100 people died. So the message here, yes, mycotoxin could be very dangerous sorry, if the toxin produced inside the food in large quantity. In fact, this report indicates that the corn was used to make bread. Sorry, and people who consumed the bread, 100 of them died. Usually mold found in different food products, especially cereal and pulses. And because mold loves dark environment, moist environment, so if the storage area is very humid, mold can produce and grow and produce toxin. And also, another way, as we'll discuss later on, if this product offered to animal as feed, the animal will feed on the contaminated grain or corn or cereal, and they may excrete the toxin in the milk, especially when we talk about bovine milk. So, generally speaking, as we discussed with bacteria, we spoke about infection and intoxication. Again, when we talk about mycotoxin, there are two major ways where mycotoxin can reach a human being. Namely, it can cause direct contamination while the food is contaminated with the mold. When you eat the food with the mold can produce the toxin and cause intoxication. So this is one way, food or feed sample contaminated with the mold itself. The second way, indirect contamination, the ingredient will be contaminated with the toxin produced by the mold. So you can use similar analogy to infection and intoxication. So indirect contamination is related to infection, because in infection we said whole cell will be contaminated the food. And here we are talking about the mold itself contaminating the food or feed. So indirect is similar to infection, while intoxication is similar to indirect contamination, because indirect refer to the ingredient being contaminated with the mycotoxin. So the ingredient contaminated with the mycotoxin, we call it indirect. If you want to apply analogy, it is similar to intoxication. But no matter if it is direct or indirect, the outcome could be the same, meaning food poisoning. Most mycotoxins are quite resistant to most heat processing conditions. So we said before that bacterial toxins are very heat resistant. If you compare bacterial toxin to mycotoxin, again, bacterial toxins are more resistant than mycotoxin. But mycotoxin, if you apply severe heat treatment, you may be able to destroy and eliminate mycotoxin. This table shows you different type of species. I'm talking about different type of mold and the type of toxin they are produced. Again, I don't expect you to memorize this. Just keep in mind that when we talk about mycotoxin, we distinguish among 4 or 5 major mycotoxin, usually named B1, B2, G1, G2, and M1, M2. Sometimes they simply mentioned M. And the distinction or the variation among all of these, usually toxins are analyzed using gel electrophoresis, meaning it is separated using special gel in a buffer solution and current of electricity. So when you separate them under these conditions, the B1 and B2 are very similar, except the molecular weight is different. From memory, I think the molecular weight for the first one is 314. No, this is 312, and B2, 314. So it varies only by 2 grams in terms of molecular weight. And in fact, the chemical structure varies. It contains only 2 extra hydrogen. Why do we call it B1, B2, G1, G2? When you separate the mycotoxin using gel electrophoresis and examine it under UV light, the B1 will appear blue in color, while the green or the G1 and G2 appear green in color. And that's why they call it B1, standing for blue, G1 standing for green. So the difference between the 4 is very minimal. It is related to the molecular weight, but the reason it is called blue or green is related to how does it appear under the UV light when you separate them using gel electrophoresis. So this will cover B and G. How about M? Why do we call it M? M, in fact, is related to the metabolism. So as I mentioned before, if we have grain infected with mold offered to the animal feed on the grain infected with mold. So the grain may contain B1 and G1. So when the animal digests the mold B1, G1 or B1, B2, through metabolism, the mycotoxin will be excreted in the milk. So when The mycotoxin isolated from the milk, we call it M-toxin. So rather than say toxin generated through the metabolic reaction, it is abbreviated as M-toxin. So I repeat again, B-toxin, it is called because it appears in a blue color when we isolate the mycotoxin using gel electrophoresis. G, because it appears green. M, because it is the result of the metabolic hydrolysis of B1 or B2. So if it is coming from B1, we call it M1. If it's coming from B2, we call it M2. So among all of this, usually B1 is very predominant compared to B2, G2, and the final product of metabolism, M1. And that's why when you read in the literature, usually they don't mention M1, M2. They simply list M, standing or meaning it's coming from B1. Aflatoxin can produce acute necrosis, cirrhosis, and carcinomia. And here we are referring to infection or dead cell in terms of necrosis. Chrysosis infects the liver of the human being or causing cancer in the liver of the animal or human. As you can see, the LD50, as we defined it before, the dose required to kill 50% of the population is relatively speaking very small when we talk about mycotoxin. It is from 0.5 to 10 milligram per kilogram body weight. So if you are talking about, say, 10 milligram per kilogram, if a person weigh average 100 kilogram, which is unlikely. So a 100 kilogram person will need only 1 gram mycotoxin to be killed. But if we say on average the person weighs 50 kilogram, it is only 0.5 gram can kill that person. So that's what we mean by these values. So it is, relatively speaking, not as dangerous as bacteria, but it can cause or kill the targeted person or animal. So that briefly the mycotoxin. I mentioned at the beginning the 3 major categories of toxin are bacterial, algal, and mycotoxin. We did mention viruses, but viruses usually do not produce toxin. They are agents which cause diseases directly. So the following or the remaining slide talk about diseases caused by viruses and some worm or protozoa that we discussed before. So I'll go through them very quickly, and you don't have to pay too much attention to these because they are not as critical as the last 3 groups, meaning bacterial, algal, and mycotoxin. We did mention viruses and their importance before. We all know that when they're present in our food sample, they remain dormant, but whenever they move from the food to the host, they start to reproduce very quickly and cause a problem. We knew that viruses are very resistant, but using sanitizer or sanitization, again, there was another question during the break about sanitizer and sanitization. Remember when we said sanitization and sanitizers, we are talking about chemical used to sanitize or to make the surrounding environment sterile or free from microorganisms. And the best example used in the food industry, hypochlorite, for example. Usually they use it at concentration not to exceed 200 parts per million or ppm. Peracetic acid, which I mentioned before, it's a combination between hydrogen peroxide and acetic acid. And we have ethanol, like in the lab, we use ethanol 85% as a sanitizer when you clean the bench top. So you are doing sanitization to your working station. In terms of viruses, we distinguish between hepatitis A and E and Norwalk virus group. And again, they are not dangerous if we apply good hygiene and sanitation practices, because sanitization can eliminate them. Similar to what we discussed in terms of infection and intoxication, you can read through the source, the symptoms, and how do we control it. So I'm not going to ask you about these details. You can read through them. Again, our focus should be on bacterial, algal, and mycotoxin. The parasite, which is the last group, again, it is not as dangerous as bacterial, fungal, or algae. Anyone remember why? They can be easily killed by heat or even by freezing. Remember, we said bacteria cannot be killed by freezing. When we talk about parasites, because they are large in volume, in mass, because they are large in size, even during freezing, the formation of the ice crystal will lead to the rupture of the cell wall and cause death. Similarly, because they are large in size, they can be easily killed by regular heat treatment. And that's why in most developing countries, we don't have problem related to parasites in food, but some country with bad hygiene and sanitation practices may still suffer from these type of parasites. And we discussed before, I believe, lecture number 3, about the different group from roundworm to tapeworm to fluke and protozoa. And we did mention that some of these, like Trichinella or Giardia as a protozoa, could be a problem. Remember, I mentioned the example about someone threw dead dog in the water reservoir in Sydney a few years ago, and consequently the water became contaminated with Giardia. But at that time, people were advised to boil the water before they drink it, because boiling can kill Giardia very easily. The other example I mentioned before when we spoke about this protozoa is Trichinella spirolysis under roundworm, Trichinella spiralis, because it is associated with pig's meat. Again, most developing countries, they apply good hygiene and sanitation practices in the farm. And if they apply good processing conditions, there should be no danger. But in the history, there have been some report about some foodborne illnesses related to Trichinella spiralis. So you can read through this slide. I'm not going to ask you a question about them. We move to the next lecture, which is microbial identification. Before I start, any question again about what we have discussed in terms of microbial hazard, toxin, bacterial toxin, algal, fungal? So in terms of microbial identification, and in fact that's what you are currently doing in terms of identifying what type of microorganism you isolate from food sample, usually the focus will be on pathogenic, not as much as when we talk about spoilage microorganism. And again, the reason for that, pathogen are the disease-causing microorganism, like when we talk about Salmonella, Clostridium, Bacillus, and many others microorganisms. So when you are in charge of food safety and if you want to identify what causes the food illnesses, it is usually recommended that if you can narrow down the name of the microorganism up to the strain or even the species, it will be good because, again, the more you narrow the identification or the identity of the pathogenic microorganism, the easier the treatment of that foodborne illnesses. Saying that, spoilage microorganisms, again, are important, but because they are not pathogenic, they become second on the list. But saying that, we have to be careful that whenever there is a spoilage microorganism, there is a chance that it is associated with pathogenic microorganism. And the reason for that, as I mentioned before, usually when the microorganisms grow, destroy the tissue, they create better environment for pathogenic microorganisms to emerge. Like, for example, when we talk about proteolytic microorganism, if the spoilage microorganism destroyed the upper layer of the, say, meat sample, the proteolytic microorganism Clostridium will emerge immediately. So we have to pay attention no matter if we are dealing with spoilage or pathogenic microorganism. Again, as we mentioned earlier, we always talk about pathogenic spoilage microorganism and the need to identify this microorganism to prevent spoilage, to prevent disease. Similarly, we said that microorganisms need to be identified because they are widely used by the pharmaceutical industry, by the medical industry, by the wastage or the waste treatment industry, in the dairy industry in terms of fermented dairy product, in the pharmaceutical industry in terms of the production of different type of enzyme and hormone. So I listed here, for example, using pure culture as a starter culture to ferment milk, to make a cheese, yogurt. We talk about probiotic bacteria or probiotic microorganisms, which are classified as good microorganisms for our gut. I don't know if any of you watch the news last week on the current affair. There was a talk about new research in Israel. A group of scientists were examining probiotic bacteria. In this drink, we call it Yakult. And they mentioned that the general trend which has been reported in the literature about the health advantage of Yakult is incorrect. So it's spelled Yakult or Yakult. I'm talking about like 100 milliliter or 150 milliliter container sold in the aisle of the supermarket, very sweet drink. They say that Bifidobacter in this Yakult can kill the good microorganism in our gut. While generally speaking in the literature, it has been reported that it is good, and that's why they encourage kids to drink it on daily basis. My advice usually is not to drink this because it's very sweet. Simply eat normal yogurt because it's very rich in lactic acid bacteria. When we talk about microbial identification, again, we have to keep in mind about the difference between strain, species, genus, family, class, division, kingdom, as we discussed in the first lecture this semester. But generally speaking, for our purpose, it is easy to identify the microorganism up to the genus or even the species. Strain, it will be difficult because strain identification require some additional serological test or molecular microbiology analysis of the DNA or RNA. But using the biochemical test, you can narrow it down up to different species. So when it comes to the microbial identification, there are traditional method as well as rapid method. And the traditional method, as you can guess, the basic one is the microscopic examination, and that's what you have been doing for the last few weeks in terms of gram staining, lactophenol staining, and distinguishing the morphology in terms of shape, size, and the color under the microscope. This reminds me with the question in the mid-semester break when I asked the question, compare between gram-positive and gram-negative and explain how this variation affect the result of gram staining. In fact, some students misinterpreted the question and they said simply gram-positive appear blue, gram-negative appear pink under the microscope. So when you read the question carefully, it said compare between the chemical composition of the cell wall of a gram-positive and gram-negative and explain how it affects gram staining. So I'm not asking you about what is the color of a gram-positive and gram-negative. I'm asking you to analyze the chemical composition, compare between the peptidoglycan, the lipid layer, and some of you did an excellent job. Some of you answered it perfectly. Say gram-negative contain more lipid, and consequently, during washing with the organic solvent, it will create hole, and that lead to the leaching out or losing the crystal violet iodine. And when you add safranin, it will be picked up by the gram-negative bacteria. How do microorganisms appear under the microscope? This is just illustration of different shape. This is, again, not under the microscope, mostly on the Petri dishes. So you have seen all these type of shape, smooth, circle, knob shape, oval shape. And remember, we noticed this oval or ellipsoidal with yeast, very often with the yeast. The filamentous with the mold. You all have done gram staining. And again, just briefly here, if you are taking microorganism from McConkey or brilliant green, you would expect that you are after gram-negative bacteria. If you are getting gram-positive, it is more likely that malfunctioning in gram staining. It could be the steps when you add alcohol or acetone, or it could be the time. So the traditional method of microbial identification, remember we start from growing or isolating the microorganism and start preparing what we call it pure culture. And then in the pure culture we use either differential medium or we use selective medium, such as brilliant green, McConkey, which is classified as selective medium because it contains crystal violet, which prevent the growth of gram-positive bacteria, but encourage the growth of gram-negative bacteria. We used Oxford Listeria to isolate Listeria monocytogenes. Some of you used Baird Barker agar because it has special chemical which turn black when you have a Staph aureus. So you have been through this process in terms of isolation and preparation of pure culture. Then we move to the biochemical test, which depend on changes in color, changes in pH. Like when you have brilliant green or McConkey, most of you notice changes from pinkish to yellowish color. which lead us to distinguish between fermenter versus non-fermenter. Like fermenter microorganism will hydrolyze the sugar, increase the acidity, lower the pH, and consequently the bromophenol in the media will change color from pinkish to yellowish. We assess the production of gas. Remember, you used catalase test and watched for the air bubble. If you want to go deeper in the analysis, again, you can use serological test. And the serological test is usually based on the interaction between antigen, antibody. So you have to use either the ELISA technique or any other similar technique. So you isolate the microorganism and you treat it with a special targeted antibody and see if it is positive or negative to a special serological test. phage typing, again, another advanced technique for microbial identification. Remember, we discussed phage typing before when we spoke about viruses, and we said basically it is based on the principle if you have a media spiked with the microorganism, and in this case it has to be bacteria. We don't use it with mold or yeast because viruses are very specific for bacteria. Then we add special viruses. So this represents the bacteria, and this represents the virus. And we said if this bacteria is specific or this virus specific to this bacteria, it will create clear zone. And the creation of that clear zone is, in fact, what we call it the phage typing. So we identify the bacteria using specific targeted virus. So if the virus did not hydrolyze the bacteria, meaning this bacteria is not specific to this phage. Phenetic identification of microorganism, and when we use the word phenetic, in fact, we are talking about the morphological characteristic. And when we talk about the morphological characteristic, we name the color, the shape, the size, being motile or non-motile. Like we have a special microscopic slide. So in the middle of the microscopic slide, you have a cavity. So you put your microorganism in and you watch if the microorganisms are moving inside your sample or not. So we distinguish between motile or non-motile. And this, of course, related to the presence or absence of what we call it flagella. Remember when we discussed the microbial cell wall, we mentioned some bacteria contain flagella, like long thread, and these flagella help them moving around. In fact, Berge's manual is a common manual. It has been around for many years, and it is mostly built on the phenetic identification of microorganism. Like, I would strongly advise you to visit the library and check for Berge's manual if you are identifying your isolated microorganism. So it starts from the basic being gram-positive or gram-negative, then cocci or rod shape, motile, non-motile, fermenter, non-fermenter. Then you can narrow down the isolated microorganism into the level of species or genus, depending on how many criteria you can collect. What are the advantages and disadvantages of conventional method? And when we talk about conventional method, basically we are talking about the technique that you have been applying during the last 8 weeks, from growing the microorganism, purify the culture, take the culture and test it using biochemical reagent. So if you are asked question about the advantage, disadvantage of conventional method, just remember what you have done. So you can call, it does not require any specific equipment like PCR to isolate the DNA. We didn't use PCR machine. We didn't use gel electrophoresis machine. So it is very basic, very conventional. It is cheaper than the advanced method. But in terms of media, we use a lot of media, we use a lot of chemical, we consume a lot of time. It has been few weeks and you have been still working on the identification of these microorganisms. Limiting factor in the detection and determination of the microorganisms sometimes related to appropriate legislation. Like if you read the FDA regulation and even the Australian New Zealand Food Authority, they say when we talk about salmonella detection, the method of analysis should be able to detect at least 1 bacterium in 25-gram sample. So when we talk about salmonella, the method should be able to detect at least 1 bacterium. And you recall we said bacterium is singular, bacteria is plural. Because I'm talking about one, we say 1 bacterium of salmonella in 25-gram sample. So to meet this regulation, you cannot use, for example, 10 gram for the analysis. Because 10 gram is less than 25 gram. So in another word, because of this legislation, when the sample is analyzed, you have to use at least 25 gram. And to play safe, in fact, you better use even 50 gram rather than 25 gram to meet these guidelines. So sometime with this regulation, conventional method could be a problem. Like sometimes people with some of these conventional method use 1 gram sample plus 9 milliliter pactobiptone to save time and money. Meeting the regulation guidelines, sometimes it is very tricky. You have to make sure that you use the appropriate weight of the sample. And in fact, that's the reason in our practice in the lab, you always advise to use 25 gram plus 225 milliliter peptone. Again, some of the, if you call it disadvantage or minuses about the conventional method, the results are uncertain. Like some of you may have concluded, yeah, it is gram-positive rod, but is it Listeria or is it lactic acid bacteria? Because both of them are rod and gram-positive bacteria. So even if it is lactic acid bacteria, is it Lactobacillus bulgaricus or Lactobacillus cremoris? So we need to do further serological tests to be able to break it down into strain or subspecies. Some microorganisms may take weeks to grow. Again, under conventional method, we basically using bacteria that can grow, relatively speaking, very quickly, like lactic acid, Listeria, gram-negative bacteria. But when we talk about Mycobacterium tuberculosis, Legionella, it takes weeks to grow this microorganism. By the way, Legionella is a common bacteria which infect the cooling and heating system. And from time to time, we talk about Legionellosis, similar to salmonellosis. People can catch salmonella through the cooling system or through the heating system because this microorganism can grow in the water reservoir, which is used for cooling or heating. Difficult to characterize or both. So, and in fact, this statement depends on what type of microorganism you are dealing with. Like if it is yeast and it appears ellipsoidal in shape under the microscope, definitely it is yeast. But if it is rod gram-positive bacteria, it is hard to say what exactly that bacteria. Or if it is rod and gram-negative, it is hard to say, is it Salmonella, is it Klebsiella, is it Shigella, because all of them are gram-negative. So these are some of the minuses into conventional method. Advantages or positive aspect of it, if you call it, require little capital investment. As I said, we don't need PCR or any sophisticated equipment. Expenditure on consumable is not expensive, especially if you try to prepare the media in the lab yourself. Currently, we use the media preparation unit because of large number of students, but still, relatively speaking, very cheap in comparison to the other technique, as we'll discuss in the following lecture the week after the mid-semester break. It is, generally speaking, labor intensive, meaning you have to spend a huge amount of time in preparation. You have to stand in the lab for a couple hours to isolate the microorganism, count them, prepare pure culture, and so on. So I'll stop here and we'll continue after the mid-semester break. Anyone has any question?